Okay, good morning, good afternoon, dear viewers, friends, colleagues. You are here on the channel Innovative Sustainability. You can see all the different aspects of sustainability presented here. Today we have our speaker, Dr. Rand Elshereef, and he will be talking about sustainability in the pharmaceuticals, focusing on the process efficiency. I would like to introduce our speaker. I know Rand from my Sanofi times. We worked together on several projects. He got his master's degree from the University of McMaster in chemical engineering, then followed by a PhD degree in chemical engineering at the University of Waterloo. Then his career started as an instructor at McMaster University, then postdoctoral researcher and engineer at the company called Prosensus, then principal engineer at Bayer, and then deputy director, specializing in advanced data analytics at Sanofi. That's where we worked together on several projects, and Rand introduced me to the world of data analysis and modeling. And so it was a great time to work together on those projects that helped me a lot. And now he moved on, and he's now sessional instructor in W. Wolf School of Engineering, Practice, and Technology. And as I said before, he will be talking today about the data analysis, how to make the process more efficient. Rand, the floor is yours. Welcome to the channel. It's a pleasure to see you here.
Thank you very much, Marina, for the introduction. And it will be my pleasure to present about the applications of machine learning and data analysis. So I'm going to share my slides now. Okay, so this is the title of the slides, Application of Data Science in Biomanufacturing. And it's presented by me. And this is the just like a background about myself. And I think Marina did a wonderful job in explaining about my background. So I am a chemical engineer, and I did my PhD in the University of Waterloo. And after my PhD, I joined a consulting company, Prosensus. And after joining Prosensus, working for 7 years in providing consulting services to chemical and pharma companies, I joined Bayer Pharmaceutical as a principal data scientist. And then after that, I worked in Sanofi as a deputy director, where my role was to extend the applications of principal data analysis towards troubleshooting and root cause analysis applications. And then now I'm working as an instructor in graduate program at McMaster University, which is biomanufacturing program as a sessional faculty. So now here we will cover these aspects during the presentation. So we start, what is data in biomanufacturing? And what types of data collected from bioprocesses? And the third part is how to turn data into actionable insights. So let's start about a general overview of what is a biomanufacturing process. Biomanufacturing process is a very large, complex process, and this is just like a general framework of a biomanufacturing process. There is some differences between different companies, between different processes, but this is an overall view of the main elements that exist in a biomanufacturing process. So biomanufacturing process, it's a process that produces vaccines, antibodies, monoclonal antibodies for therapeutic services, So it starts with production bioreactor. So we bring all the raw material, the cell cultures, media, and we start with a bio reactors, or like a fermentation reaction where we grow the cells in under suitable conditions of temperature, pressure, glucose concentration, and those cells produces the product of interest that we are interested in. So it can be a vaccine, or it can be like a monoclonal antibody, and then we have a centrifugation or filtration, where we are interested in separating the product of interest from the cell culture media. And after that, we go to a series of purification steps where we want to purify or produce high concentration of the product. And then after that, we have the final step of like a formulation and packaging, the final fill of the product of interest. So you can see here how complicated is the biomanufacturing process. So it consists of several steps. These steps are connected to each other. And imagine also the instruments that are installed in each unit operation. So this is a brief overview of biomanufacturing process. And then here we go to a definition of data. What is data? So when we say machine learning or data analysis in biomanufacturing, data is defined as anything in electronic format that can be stored, processed, communicated by humans or machines. And these data has different forms. So we have variety of forms and formats of data in biomanufacturing. The first type is quantitative data. So what do we mean by quantitative data? Quantitative data are represented by numbers, measurements, and it can be divided into two types: continuous data and discrete data. So what do we mean by continuous data? So here, when we run a kind of a fermentation or a bioreaction in a batch or fermenter, there is a start time and end time for performing this fermentation or bioreaction. And between the start time and end time, we collect data of the most important variables or measurements for the fermentation. For example, here we show profiles of optical density that represent the cell growth from the start to the end of the reaction or fermentation. Here we show the glucose profile. The glucose is being consumed by the cells growing during fermentation. You see that the glucose concentration starts high and then it drops as more cells produced and divided and produced during the fermentation. We have also like a change in the pH as well, a step change in the pH. So here we have continuous data for every batch that we operate or execute. We have continuous data collected. At the same time here, we have another type, which is discrete. So what do we mean for discrete data? Discrete data is at the end of the batch, we collect sample, okay? We send the sample to the quality control lab, and we measure the product quality attributes at the end of the batch. So therefore, we have one value, product quality attribute, per batch. So that's why it's called discrete data. However, for the other side, continuous data, we have a continuous profile of process conditions such as optical density, glucose, pH for each batch. So this is how we distinguish between continuous and discrete data. Now,
Yes, so just
the
sorry, so
second, yes,
Mhm.
go ahead.
Just to comment, basically what you're describing is if you go back to your previous slide, it refers to the first step. Like I'm just saying because in the audience we have people who understand it maybe very well and those who definitely new to the process. So what you're showing here is the analysis of the discrete and continuous data referring to the first step of the process,
Yes,
right? That's a fermentation.
exactly. So
That's
here,
all.
yeah, this is we are referring to the for
Mhm.
the to the exactly the fermentation, the first step, and of course for every step we have the same type of data, continuous data and discrete data. But this is here we are showing the data acquired from a fermentation only, right?
Thank you.
Yeah,
Yeah.
and of course. For downstream processing, we have the same, like we have continuous data and discrete data as well. So another type is the qualitative data. What do we mean by qualitative? It's categorical data. So sometimes when we look at the data acquired during manufacturing, it's not a numbers, it's just a description of the process. So we have within specification. So we can describe some batches as within specification or out of specification. So if the product quality attributes is doesn't meet the standards of quality standards, then we say it's out of specification or out of standards or poor quality. All of these types of description are considered to be qualitative, which is important for doing machine learning and data analysis. For example, classifying, if you want to find what are the contributing factors that are contributing to one batch being an outlier or being out of specification, then we acquire data and we perform machine learning and data analysis, and the data analysis will tell us about the combination of variables that are likely contributing to a particular batch being an outlier or being outside the specification. So this is how important to collect all types of data, okay, and do such type of to do analysis. Okay, so this is just like what is data and what is representative of course the same applies to other unit operations. And
Yeah, this
here,
is very important. Yeah, yeah. I just
yeah,
want to say
and
that basically a quality a test that you apply is it's like another type of test, which basically tells that batch is acceptable or not, using different terms, right? Or different types of tests, and then you refer back to the process data to link the two, and this and basically also like you said, machine learning to distinguish. Then if after you rely only on the in inline or continuous and discrete data collected, you know that it should be within the window of to make the batch acceptable
exactly,
to
exactly.
reach that kind of corridor, right, where it becomes acceptable.
Yeah, exactly. Yeah. So here the types of data collected by manufacturing in terms of all the instrumentation required for different unit operations. So here we have process data, we have also data systems, and we have PAT, process analytical technology. Process data, we have all inline data, online, offline, and at-line data. What is inline data? we will have a probe inserted inside the fermenter or bioreactor. So there is a direct contact between the inline probe and all the cell culture media and the cells broth that existed inside the fermenter. There is another configuration which is online. Online, we have a bypass that withdraw sample and flow through a flow cell. And in this flow cell, we acquire the measurements, right? So it's a kind of a recycle, a circulation here. Also, we have at-line data where we take a sample, okay, and we send the sample to an instrument that is located in the shop floor, in the same location where we have the bioreactor, okay? And we have offline. Offline is we collect a sample offline, we send the sample to quality control lab that could be existing in another building, and we label the sample, of course, and we do analysis. So this is the type of data that are collected from the process. Inline probe, bypass with online and at-line, where we take the sample, we do filtration or some sample preparation to remove bubbles or dust or whatever, and we send it to an instrument that exists in the same location. And we have on offline where we send it to a QC sample to a QC lab. Now, someone may ask, okay, what is the difference between inline and online in terms of measurements? Usually, when we have inline probe inserted in the fermenter, the measurements that we collect is subjected to an influence of bubbles and foams, and there is an interference, especially when we have fiber optic probe that acquire for example, the fluorescence or spectroscopic measurements. If we have like bubbles, those bubbles will cause light scattering, and this will affect the measurement that we are collecting. Therefore, we need to have some machine learning algorithm in order to clean the data and make sure that we remove any anomalies or artifacts from it. However, for online measurements, since we are applying a bypass and we collect a sample through a bypass, okay, this bypass is less subjected to the artifacts that we saw for the inline measurement. So there are a couple of advantages and disadvantages for these different configurations, and then we go to the next step, which is start with these data systems. So we have two different data systems that exist in pharma industry. MES, manufacturing execution system. So manufacturing execution system, we have information about the production, scheduling, the work order, tracking number. So whenever we have an order from upper management to produce like a particular quantity of the product, then an ID number or our order number is assigned to the future batches, and all ID number pertaining to this particular batch will be assigned to all raw materials that goes into the batch and all a kind of intermediate steps that goes to the batch. To that particular batch. So this is related to traceability. How can you trace a batch, and how can you identify all the raw materials that goes into a particular batch by tracking those, having an order number or tracking number. And also the second one is lab information management system. So I would like to add that MES system, it will help us in scheduling and managing all the unit operations in terms of demand, large scale management of production information. Now, the next step is one is lab information management systems, which is LIMS. Here we have information about all the analytical tests that we perform for like product quality attributes, protein or antibody aggregation behavior, the bioassays, the sampling tests, the calibration required for each, and the instrument calibrations information. It has a large information about all of these types. And we mentioned before that one technique is offline measurement, where we withdraw a sample and we send it to the QC lab. And here we just like we run measurements and tests on these samples offline. And here in lab management systems, it will help us to arrange all of the information where we have the information related to each lot measured in and arranged in a suitable format for analysis and for monitoring. Now, the last one is advanced techniques. We have advanced such as process analytical technology. So we have here 4 different types: Raman, NIR, ultraviolet light, UV, and FTIR, Fourier transform, infrared. Those types are based on measuring the optical properties of the cell culture media. So here we can acquire like a spectrum that represents all the thousands and thousands of species that exist in the cell culture media. And we can acquire Raman, NIR, and UV measurements in real time, okay? And this will represent all the chemical information. It will capture all the chemical information and biological information of what's going on in the fermentation during the time evolution, from the start of the batch until the end of the batch. So this provides a multi-dimensional data in order to understand or capture what's going on in the fermentation or during the bioreaction.
Yes. So if I may, yeah, I think it's a great slide because first you introduced types of probes linked back to the types of data you just described, like continuous, discrete. So here we're talking majority of cases about continuous when you're saying inline or online probes, right, and sensors such as PAT. And on the other hand, how it all comes together, and when you said that it's a tracking system, when you have the same number assigned that relates to the batch, which will be produced to all raw materials. So it's all like elements of GMP that, also, becoming aware for the viewer that this is really GMP talking. It's basically encrypted in the process. And, when you do it, you kind of learn as you go. But this is a great example where, you kind of highlight and emphasize. Well, the system tells me that it's about to stop the meeting at 5 minutes. So for the interest of time, maybe we can stop here and then we'll start another session just to the viewers. Another session will be continuation of this presentation, and Dr. Rand Elshereef will continue and complete, to the next steps where how the data is analyzed and how it's used. Okay, so we'll see you shortly in a few minutes. And right now we'll stop. So long. Please stay tuned. Thank you. So I will disconnect now and we'll see each other soon. Okay, thank you.
Okay.